Welcome. In this video, we're going to talk a little bit more about classical conditioning. So by the end of the video, you should be able to identify the area of the brain that's important for classical conditioning and the area of the brain that allows the behavioral response to occur. Now, Pavlov, who was the one that discovered classical conditioning and all the principles of classical conditioning, he really felt that this conditioning strengthened connections between different centers in the brain. So the idea is that we had a conditioned stimulus center and an unconditioned stimulus center and an unconditional response center, and that these two were connected. The UCS and the UCR were automatically connected. They had a strong connection. So every time that this area was stimulated, this created a response, the behavioral response here. And that there was a weak connection here perhaps, and that over time with those pairings, this weak connection became stronger. And classical conditioning is a type of associative learning, where you're learning to associate two different stimuli. And so that's why it's called associative learning. Now, Pavlov discovered classical conditioning back in the late 1800s, 1890s, early 1900s, somewhere around then. And then Carl Lashley came along in the 50s when we were just starting to distinguish the different types of memory. And he wanted to search for the engram or the memory trace. And so he had animals run in mazes, and then he made cuts in their cortex, and he never identified the engram or the memory trace for memory because he was thinking that all memory was the same, that it was all going to be located in one area of the brain. And then fast forward to the 70s, and Richard Thompson searched for the engram. And that there's a video that he goes into in detail what his research involved. But I just want to remind you that Richard Thompson was very important in identifying the engram or the memory trace for classical conditioning. So again, this search for the engram or the memory trace, which is a physical representation of what had been learned. And as you can see in the other module link, or the other video that's linked in the module page, is that he used rabbits for this, to study this classical conditioning response. And I actually had friends when I was at USC, so Richard Thompson was at USC when I was there, and he had this big lab, and he has what we call minions kind of all over the country doing research similar. Some of it's fear conditioning, some of it's other type of learning research, but in his lab, they worked mainly on rabbits for the eye blink response that we'll talk about on the next page. So Richard Thompson's search for the engram suggested that for classical conditioning, it was located in the cerebellum. And so this is a diagram of the paradigm that he used. And so basically you had the rabbit connected up to a contraption which can measure the eye blink response. You can do the same thing in humans. They would give a puff of air into the eye, and the puff of air naturally causes the animal to blink its eye. And so here you can see tone on, air puff, and then over time the animal learns to blink its eye in response to the tone by itself and not just the puff of air. So the area of the brain that he localized the engram to was the cerebellum. So again, in his search for the engram, it suggested that the cerebellum was going to be the location. And so here we have a rat brain, but the cerebellum is very similar to ours. And in particular, the lateral interpositive nucleus was the location that he found was responsible for learning the classical conditioning paradigm. And so we'll talk a little bit about, on the next couple of slides, about how he discovered this. The red nucleus is another area, and we'll learn that this is responsible not for the learning process, but for the behavioral response itself. So we're looking at the cerebellum, the red nucleus in the midbrain, and then projecting here to the sixth cranial nerve. So let's break down how Thompson found this memory trace for classical conditioning. This is the learning curve for the eye blink response in an intact animal. So an animal that has not had anything done to its brain, just the normal response. And this is the percentage of conditioned responses. This is the number of sessions, so the number of times that you had that tone paired with the puff of air. And you can see, and of course, we just blink our eye randomly, right? And then over time that increases. And then you kind of have this plateau. This is from the actual paper, the peer-reviewed paper that was published. I like to see a more realistic would be a more curved version of it. We'll see that at the end, but just keep in mind that there's this gradual learning process, and then eventually we kind of plateau out. And that is just what a normal learning curve looks like. What Richard Thompson's lab observed is that there were changes occurring in the lateral interpositive nucleus of the cerebellum. They knew that there were changes, and they wanted to know, is it responsible for the behavioral response? Is it responsible for the learning aspect of classical conditioning, or both? And so they've done a variety of different experiments, but in this one that we're talking about here, again, they wanted to know, is it responsible for learning or the response? They inhibited the lateral interpositive nucleus. So you can do this in a number of ways. They had some studies where they actually removed it. I believe that's what's talked about in the video linked in the module page. But in this one, they're inhibiting it. So they're not damaging the lateral interpositive nucleus in any way. You can do this with drugs. You can do this a variety of different ways. But what they found is, in this second graph here, you can see when they inhibited the lateral interpositive nucleus, so inhibited means it can't fire any action potentials. Here's a percentage of conditioned responses, and you can see here over sessions, there's no learning that going on. They're not producing any behavioral response, regardless of how many times that tone is paired with a puff of air. Now, when you remove that inhibition, the animal can learn. But as you can see here, so think of this is animal number 1, and these two graphs being animal number 2. So animal number 2, lateral interpositive nucleus suppressed. Session 1, 2, 3, 4, 5, 6. They let that lip recover. So this would actually technically be session 7. So if we were learning, we'd be all the way up here, right? So the learning response would be up here if they had learned when the lateral interpositive nucleus was inhibited. But it's not. It's down here. It's a normal learning curve. It looks very similar to what you see here. So the comparison, like I said, this would be nicer if it was a little bit more smooth, but you can see the similarities in the two, hopefully. Removing this, or removing the inhibition and seeing this normal learning curve suggested that the lateral interpositive nucleus of the cerebellum was necessary for the learning and retention of the eye blink response. That area of the brain within the cerebellum was necessary. It's needed. The lip is needed for the learning of the classical conditioning eye blink response. And so that's just showing you, can here, reminding you of where the lip is, showing you this would be the 3 graphs that we've looked at so far, 1, 2, and 3. Now, another area that they saw some activity in was the red nucleus. So they knew that there were changes in this red nucleus. The question is, again, is it responsible for the learning or the conditioned response? Maybe one area wasn't responsible for the learning, maybe it wasn't just the lip. So again, we had an animal with, that was intact. And then they also had some studies, again, like the video linked in the module pages, where I believe they actually removed the red nucleus. And then here, to make it a little bit better, instead of just removing it, because then you can't see what happens afterwards, they inhibited the red nucleus. So again, using some sort of drug, you can inhibit the red nucleus so that it doesn't fire action potentials. And again, you see no eye blink response. So this is the animal here when the red nucleus is suppressed, again, not firing any action potentials. This is the percentage of conditioned responses, the number of sessions. And you can see here, it's even less than you saw when you inhibited the lateral interpositive nucleus. Now, what happens when you remove that inhibition? So this is the graph when you remove that inhibition. So again, imagine this is animal number 1 here intact. This is animal number 2 in the second 2 graphs. And so here we have session 1, 2, 3, 4, 5, 6, 7. And the number of conditioned responses is way up here at 80%. That's pretty similar to what you see in that plateau of the normal learning curve. So this suggests that learning occurred somehow during this time, even though there was no conditioned response. The animal was learning something here. And once that inhibition was removed and the behavioral response was allowed to occur, you can just see that the animal learned. No learning needed to occur anymore because it had already learned to blink its eye in response to the tone. So this suggested red nucleus was not needed for learning, but it was needed for the behavioral response. Whether it's the unconditioned response or the conditioned response, that red nucleus was needed. And it makes sense because it does project to the 6th cranial nerve. And again, here's just another summary of all. So the summary of all this. So the idea is that your lateral interpositive nucleus is projecting to the red nucleus. Okay. And then we also know that the red nucleus stimulates this 6th cranial nerve. So this is going to be your unconditioned stimulus response pair. This is going to be your conditioned stimulus response pair. So this is a weak connection, and as you pair those, this becomes stronger. So now this, stimulating this, can elicit that eye blink response. So stimulating the lip then now stimulates the red nucleus, which allows it to cause the eye blink response.